Hi, everyone. My name's Sebastian. I'm a degreeless heathen working out of a home lab in Huntington, New York. And I'm the host of Tears and Rain, a podcast focused on the stories and human elements behind the process of getting a PhD. So today in episode 4, we have Lev. So please take it away. Introduce yourself and tell us your story.
Yeah. Hi, I'm Lev. I'm currently a postdoc. Did a PhD that was heavily impacted by the pandemic. I'm a microbiologist, and I just wanted to also say that if you're a heathen, may we all be more more sacrilegious. um, yeah.
Very cool. So tell us a little bit about how you got to deciding you want to go into grad school. That's, I guess, one of the
Yeah.
first questions. Yeah.
Yeah. Well, so there are There are a lot of ways that I could answer that question. A very, I guess, personal history way of doing it is that, my family immigrated to the US when I was 6, and in my family culture, there's both a real value on getting as high of an education as possible, but also, this is something that I've been trying to really get away from, but a real fear of not living up to potential, and that, a kind of moralizing of this perfectionism, which has been kind of harmful to me, and I've spent a lot of time in therapy getting away from it. But the point is that by the time that I was going to college, I felt really compelled to try to do the most ambitious thing that I could. Like if there's a possibility to do something, I felt that I needed to pursue it. And so I went to college with the intention of doing an MD PhD.
Wow.
Uh, and uh, I left that path for a couple reasons. One is I came to feel that I didn't want to, nor could I be a doctor. What just largely on an emotional level, I felt that I wouldn't be able to handle the pressures and quandaries of like denying people care because of their medical
Mm.
insurance and like all these different things
Ouch.
that I just felt I didn't have the fortitude for. And I ended up joining a lab during my undergrad. I was studying this weird little organism called Tetrahymena thermophila, which is one of the protists, these single-celled eukaryotes. And I just was surprised by how much I loved it and just this real kind of whimsical joy of working with a strange organism and being delighted by what it does. I remember there was one day that I remember very clearly being in the lab and preparing the culturing medium for Tetrahymena and thinking that it smelled delicious. And I was like, wow, if I think this smells delicious, then I guess I really like being here.
Wow.
um, yeah, and so I feel like I got extraordinarily lucky with that lab and with my undergrad advisor, who really cultivated a a kind of peaceful joy, I guess, of doing research. And so I decided to pursue a PhD. And I didn't really know, I knew that I liked microbes, and that, I would never want to work with animals, and that, plants are probably going to be a bit too slow for the sort of thing that I wanted to do, um, which I
No
feel a
offense
bit
taken.
different. Yeah, I feel a bit differently about it now, but back then I was like, oh yeah, doing quick experiments feels better. And then I just like I feel like my trajectory is always characterized by trying to ask as many people as I can for advice and stumbling on someone who understands me and then it working out. And so I spent a while asking all the professors who I liked at my undergrad, what they thought I should do, and some of them gave me ridiculous answers, and some of them gave me ones that seemed reasonable. Um, and I took a gap year. Uh, I stayed in the same lab, but worked with a totally different organism, with one of the diatoms, which are also single-celled eukaryotes, but they're photosynthetic. They have these silica shells that they live inside of. Are very beautiful, difficult to work with, um, and ended up deciding between Berkeley and Caltech for where I did my PhD. And I think in the end, it felt like the sorts of labs that were there, the sorts of people who I got along with, were very similar, but the sizes of the two schools were very different. And so I ended up being drawn towards the smaller school, in part because I really enjoy teaching, and I thought that I'd have a more fulfilling experience working with smaller class sizes than, being a teaching assistant in a 400-person lecture course or something like that. Um, and that's how I ended up at Caltech for my PhD. We have the rotation system, so I did 3 rotations, one of which I cut short because it was bad. I wasn't having a good time at all.
Could you tell us a little bit about what was bad, if you don't mind?
Yeah, yeah. I mean, this can get a little spicy. I guess people might be able to figure out what lab it was, but I won't say any names. I felt like the lab simply wasn't set up to do rigorous work.
Hmm.
It's this interesting thing where, on the one hand, there was a lot of this kind of whimsy, almost reminiscent of like a early 20th century, like 19th century embryology of like,
Hmm.
you know, uh, the whole world is out there to ask interesting
Hmm.
questions about, and we can try these off the wall experiments and get something interesting out of it, which was really interesting because it feels kind of anachronistic. And I felt that I could learn something about thinking about science in a way that's kind of lost. But I feel like that has to be counterbalanced with a kind of rigor that was missing. And I felt just a lot of cognitive dissonance about how the work was being done in that lab, and
Hmm.
didn't have a good time.
How did you because that's a very experienced perspective. How did you come about having that thought? Like, was there something that preloaded that with your prior experiences? Because it's hard to sometimes know
Yeah,
what's a good lab, what's a bad lab. Bad
yeah,
lab.
yeah,
Yeah.
yeah. I think that's a very important question. And it comes down to me working in several different labs and getting a sense for it. So, the very first lab I worked with, this was over a summer in Colorado, where I wanted to go to be closer to home. And it was kind of funny. This was an oncology lab, run by an MD PhD, and one of the it was basically 2 people and me, one like 2 staff scientists, and the staff scientists kept fighting over me in terms of how I should do the RNA extractions, where one of them thought that I should use a robot that they had to do the RNA extractions, and the other one saw himself as a kind of modern day John Henry, fighting against the steam engine, and that everyone should do things by hand. Um, and so that taught me about how to recognize just kind of a passive aggressive lab environment, but which was a great lesson, but extremely stressful summer. Um, and then the main lab that I worked in during my undergrad, there were a couple of things that really stuck with me of what my advisor taught me. His name is Aaron Turkowitz. One of the things was just an offhand remark during a lab meeting when one of the postdocs was presenting some data. And she presented some microscopy images and said that, oh yeah, this means this. And Aaron stopped her and said, look, when you say things, you start believing them.
Ooh.
And this is something that we don't know, so you shouldn't say it. And it was such, I don't know, such a clarifying moment of an intellectual honesty and like introspection that just making a claim or an interpretation makes you believe in it.
Mhm.
Like allowing yourself to make a declarative statement in that way. And that stuck with me, not just in science, but overall in my interactions with the world. And so I think that working with Aaron in his lab, it's kind of like, being in a good relationship, as in like a very good friendship or a very good romantic relationship, where you learn the standards for how you want to be treated, how you want to
Mhm.
work with somebody. And so when I came to my PhD, I felt like I had, if not an understanding, at least like a reflex that something is wrong
Mm.
when I encountered it.
Yeah, that makes a lot of sense. And so for folks who are just starting in their rotations, is there any advice you would give in terms of like not necessarily red flag, green flag, but like things to look out for?
Yeah, yeah. I once was working on a list that I was thinking of publishing online, and then I was like, this might be bad for me to do, but because it was a little bit too obvious what labs I was talking about, I
Mm.
think. There are a couple. So one is, in my personal opinion, I think that a rotation is not about any specific outcome or any specific results, numbers of successful experiments, publishable data, or whatever. It should be about figuring out if this is a good environment for you and getting a mutual understanding between yourself and the advisor and the rest of the lab. And so rotations where you're put in a position that you're competing with someone, that's a warning sign. Sometimes it's kind of inevitable in the sense of, well, maybe the lab is popular for a good reason. A lot of people want in it, funding is tight, and by definition, there's a kind of competition. But in such situations, the lab should have enough grace and competence in terms of management to prevent it from becoming a toxic kind of competition, like with understanding that we just want the best for everyone, and in management situations there is often no way to make everyone happy. That's just the reality of things. But I've heard of labs where they're literally like tallying on a whiteboard which rotation students have gotten what done, and
Really? Oh wow.
that's just not
Yeah.
not
Especially as a
good.
student, right? Like you're just
Yeah,
starting and to like be
absolutely.
competing constantly. Wow.
Yeah, absolutely. I think that a very good sign is if you leave meetings feeling better. Like you might feel like things are still hard, that you don't understand something, that you don't know how to make your experiments work. But if you leave the meeting feeling encouraged, that's a very positive sign. That means that whoever you're talking with, be it your peer mentor in the lab or the PI or whoever, understands how to motivate you, and like how to communicate to your skills and your interests. Um, and I think that, I'm a huge proponent of the idea that you shouldn't delay your life, and that wherever you are right now, that is your life. And so when people are talking about, oh, I have to put my nose to the grindstone now so that I'm free later or something, I think is a kind of like a red herring for a
Mm.
lot of people. And so being in a lab where you see that everyone takes time off and has time for hobbies and relationships and all of that. That's a good sign too. And when it's missing, you should, take a moment to ask why that is. Like, is it just that that's what people want to do and they're happy, or that there's a kind of pressure, either implicit or explicit, that's not working well for people?
Gotcha. So with your when around your rotation, when you did you know of a topic that you were going to be addressing for your
Uh,
thesis, or how did that come
not
about?
not really. So I actually, before starting grad school, the summer immediately before, I took a microbial diversity class at the Marine Biological Laboratory in Woodsville, Massachusetts, which is like a 6.5 week long storied course. It's been around for like 50 years now. And that was really cool because, well, one, they provide financial assistance for a lot of folks. And so I was able to go for essentially free. But it's like this intensive dive into microbial diversity, primarily bacteriology, some phage work, some like amoebae and stuff like that. But I felt like going to that course really opened my mind to the world of prokaryotes in a way that I just had no appreciation for before. And so when I got to grad school, uh, one of the places where I rotated with, the lab that I ended up joining, was actually run by one of the course directors at the time for microbial diversity, Diane Newman. And there was almost like a built-in on-ramp for me in that lab where somebody in the microbial diversity course did a short summer project that was related to what Diane's lab works on, and that wasn't finished. And she was like, "Yeah, you can come do a rotation, just continue,
Hmm.
starting from that student's summer project." Um, and so that was very nice because it was something that I could have agency over. It was kind of at the right level for me in terms of my skill set at the time, and I already had some, introduction to it based on my interactions during the course. The other lab that I was considering was doing something totally different. This is Joe Parker's lab, and they study the social symbioses between ants and beetles. And so that was, a whole other world. Like we did some field work collecting ants from the San Gabriel Mountains, getting wrecked by them. a lot, a lot of ant bites, but also, really a really incredible kind of opportunity because Joe was starting his lab. We were the first rotation cohort, and so there was all this excitement about starting a new model system,
Hmm.
new lab, building everything up from the beginning. A lot of my friends, like from my cohort, who I'm still the closest to, were rotating in that lab, and so it was a very different feeling. It was this kind of established lab, more or less established project, a real predictability and stability to how it might work, versus everything is new in every respect, and the excitement that comes along with it. And in the end, when I was deciding, I posed this question to myself of, if I remember the hardest time that I had during my undergrad, and I was quite depressed during my undergrad, um, which lab would be best equipped to support me if I find myself in that space again? And I ended up choosing Diane's, and I have no way of saying like maybe Joe's lab would have been just as good,
Sure.
but given the pandemic and everything that came for it, I do feel like I made the right choice because Diane was very supportive of everything I was going through.
Wow. The that's such a tough choice going between like field adventure and established bench work, right? With
Yeah,
yeah, fascinating. I would I'm biased. I would definitely have gone with the field work just because the
yeah,
that sense of exploration is always there.
yeah,
But one
absolutely.
but you could also explore at the bench. Plenty of
Yes,
questions, right? Like diving in or
yeah,
inward versus
yeah,
outward.
yeah.
Okay, so could you tell us a bit about the formation of your project, right? Like
Hmm.
the like we can you can give us like a casual abstract of what the project might be, and then just guide
Yeah,
us through how that progressed. Yeah, especially
yeah,
your first year.
absolutely.
Yeah.
Yeah. So here's the pitch. Uh, when you're working with bacteria like the workhorse E. coli or Bacillus subtilis, Vibrio nitrogens, lots of these little guys who are very well understood. And we've optimized their growth conditions just like as far as they can go. And they have a doubling time of like once every 20 minutes. They reproduce. Vibrio nitrogens can do like 12 minutes. It's bananas. But if you think about the exponential growth rate, what you'd end up seeing is that if you just let this grow starting with one bacterium, in like 2.5 days, the bacterial colony would have the volume of the entire Earth. In less than a week, it would be the whole observable universe. And that clearly doesn't happen,
Sure.
right? Like, we're not, suffocating in bacteria right now. And so that means that in nature, bacteria are growing slowly or not at all.
Hmm.
And there are a lot of reasons for that. It might be that the conditions aren't perfect and they're starving for something. It's also stressful to grow that fast. Like you end up creating toxic compounds as your waste product, and it's fine for the experimenter to get, the biomass up very fast, but it's not ideal for the bacteria. And so Diane's lab is very interested in this idea of slow growth and
Hmm.
how to study it. And it's more difficult than studying rapid growth. It's an attempt to kind of reflect reality more closely, which is very hard to do. And it turns out that there are a lot of bacteria in the world that actually use bioelectricity, basically. During your metabolism, what we do is we take electrons off of our food and we dump them onto oxygen, and in the end that's all it ever is. There's this wonderful quote from Nobel laureate whose last name I can't pronounce, so it's Albert St. Georgi, that life is just an electron looking for a place to rest.
That's a
And
great quote. Wow.
yeah, and so there are bacteria out there that are just like putting electrons onto all sorts of stuff. There are bacteria that put electrons onto iron, onto uranium, onto sulfate, like this whole menagerie of different ways of respiring, essentially. And there's some bacteria that make their own uh, electron acceptor. So specifically, the Newman lab is interested in this group of bacteria called the pseudomonads, like Pseudomonas aeruginosa, which is a common human pathogen in chronic infections, like cystic fibrosis. And it's an interesting little guy because it's very bad at fermentation. Like it can't really do it at all. It has to respire. And so if you put it in a situation like a chronic infection where there's very little oxygen, there's very little anything to respire, you'd think that it would just die, but it doesn't. It makes a compound that it can use instead of oxygen, synthesizes it itself, and kind of uses that as an escape valve for its waste product,
Hmm.
electrons. Uh, and so for a long time now, people have been studying this class of compounds called phenazines as an electron acceptor and how bacteria interact with them and how it promotes their survival. But no one had looked into the the opposite reaction. Uh, are there microbes out there that can take electrons away from these phenazines? And the reason why that's important is if you imagine you have your bacterium and it's constantly reducing the phenazine, if it's not recycled in any way, then it has to keep exerting a lot of resources and energy to make that compound. But if there's a way of recycling it, like something takes that electron back off, then the cycle can continue
Hmm.
and you don't have to invest resources all the time to reproduce it. And so the project was to go out and take environmental soil samples and see if there are bacteria out there that will oxidize, take electrons off of this phenazine. And it's very neat because phenazines are colorful and they change color based on their oxidation state.
That's convenient.
And so, yeah, and so at the very beginning of my project, what it was having tubes that were sealed shut, flushed with nitrogen to get rid of all the oxygen, inoculated with reduced phenazine, and the one that I was using was this kind of bright green orange, bright green yellow color. Inoculate it with samples from the soil, and then see if it becomes colorless. And then if it becomes colorless, see if I can transfer that and prove that it's actually a biological process rather than an abiotic one. And that's I'd say like the first chapter of my thesis is like proving that it's biological and identifying one of the organisms that was doing it. Uh, and then the rest of the project was trying to actually figure out how this works and the genetic mechanism. And so I was developing tools to genetically engineer this isolate from the soil and then knocking out a bunch of genes and seeing if I could get it to stop oxidizing the
Classic.
phenazine. Yeah,
Yeah.
exactly. Um, and yeah, I ended up extremely lucky actually, because the first organism that I pulled out of the soil that would do this, it's called Citrobacter portucalensis, which was first described in 2017, like, 3 years before I figured out what it was. Um, and it's close enough related to E. coli that I could just adapt all the E. coli
Oh
protocols
wow.
for working with it. And
Nice.
so it was kind of the best of both worlds, where I had a soil isolate that's kind of more representative of the real environment and soils, but I had all of the E. coli
Oh
tools
my
and
god.
didn't have to
That's
bash my
so
head against
nice.
the wall.
As somebody
Yeah,
working in non-model stuff, especially for client work, I'm so envious. That's amazing. No, that's wonderful. Yeah.
yeah. I mean, it was a real stroke of luck. There was no reason for it to work out this way, but but it did. Yeah.
Very cool. Very cool. Okay, so you have that would be your first two chapters, right? Of that.
Yeah,
Yeah. And
basically, yeah.
no, I was gonna say, and then what? So you can interface with the organism. You've isolated
Mhm,
the organism. You can interface it. And what would be the
mhm.
next steps after that? What was your logic there?
Yeah, yeah. Well, so it's it took a lot of kind of iterative thinking, I guess.
Hmm.
So the first thing that I did was to figure out, not touching, the genetics at all, but what are the conditions under which this biological process happens?
Hmm.
And the very first kind of thread that I got that I could pull on was that it depended on the terminal electron acceptor that I provided in the medium. So again, the idea is that I don't have any oxygen. So whatever the organism is doing, it can't rely on oxygen as the ultimate thing that grabs the electron. But I have to grow it somehow. And so I was growing it on nitrate, NO3 minus, which is, if you look at the ranking of like preferred things to respire by organisms, it's the second one after oxygen. And I found that if I grew up this culture and I gave it neither oxygen nor nitrate, like no terminal electron acceptor, it wouldn't do the process anymore. And then I went and like step by step systematically was like, okay, well, is my reduced phenosine reacting with nitrate directly? And the answer was no. And then I was like, well, I know that when organisms respire nitrate, they produce nitrite, NO2 minus, which is much more reactive than nitrate is, just abiotically. And so then I tested if I mixed my reduced phenosine with nitrite, does the oxidation happen? And the answer was yes, but much more slowly than would explain the biological rate. Uh, and then I was wondering, I looked at the electron transport chain for this organism based on its predicted genes. I had sequenced its genome. And I found that, like E. coli, it should also be able to ferment, or sorry, not ferment. Well, it does ferment, but in this case, that's not relevant. It should also be able to respire fumarate, which is one of the intermediates in the citric acid cycle, and DMSO, which is like it's a popular solvent in the lab, but also when it's reduced to DMS, that's like the smell responsible for kind of like ocean rotten fish kind of
Mm-hmm.
odor.
Like low tide.
Uh, yeah, exactly. And TMAO, which is also kind of similar, very fishy. So DMSO is dimethyl sulfoxide. TMAO is trimethylamine N oxide. Anyway, so then I tested each of those both with my culture and abiotically. And I found that the oxidation of my phenosine, this, which I could observe by the loss of this green color, correlated with the presence of something to respire.
Hmm.
And so it seemed like if the cell is respiring anything, it's going to be grabbing electrons off of this molecule. And so that was what kind of guided me to think about all the genetic perturbations, because when you look at the electron transport chain, be it in mitochondria, E. coli, or whatever else, there are a lot of branch points where those electrons can go. But ultimately, it's all about an electron being shuttled from the cytoplasm of the cell, from NADH usually, going to these membrane-bound oxidoreductases that take that electron, put it on a electron shuttle like a quinone, like ubiquinone, menaquinone, or a cytochrome, and that electron just keeps hopping along until ultimately it gets to this terminal reductase that is coupled to ATP synthesis. And so that electron, ends up on an oxygen, the ATP synthase does a third of its turn, the protons go through and the thing kind of ratchets forward. Um, and you, so what I was trying to do was systematically introduce mutations that would break that chain at different points and see what how much do I have to break for this process to stop?
Mm.
And it turned out that this is a surprisingly generalizable mechanism.
Hmm.
Basically, for any organism that I tested, I started throwing this at a bunch of different bacteria that I had in the lab. So long as it's possible to have an electron flow through the electron transfer chain, they'll do it. And so it seems like this reduced molecule is promiscuous enough that, so if there's like almost like a voltage or a pressure that it can interact with, it will.
Wow.
Um,
Wow.
yeah,
So you're all of this seems very well laid out, and it sounds very independent. Now, uh how how confident did you feel during all of this? Like, were there points where your advisor played a heavy role in trying to help steer? Because I mean, from obviously, like hindsight is 2020, but
yeah,
yeah, I'd love your
yeah,
perspective.
absolutely. Yeah, well, so there were a couple important things, I think. One is that the pandemic started right after, like basically half a year after I did my quals, and then took 2 months to recover emotionally and spiritually. And I finally was getting back into the groove of doing these experiments, figured out the protocols, figured out everything, and then the world shut down. Um,
Oh wow.
and that was extremely difficult. I basically, I left LA. I moved up to the Bay Area to be with my now wife. We went from long distance to quarantining together, which
Wow.
is a whole other story.
Wow.
But it was not at all obvious to me how I would proceed. And one of the things that I realized after, things eased a little bit and we got back into the lab and everything was on shifts and social distancing, like once everything got stripped away from the research except for the bench work, I realized that I don't like doing experiments. Like what I like is everything that's around it. I like talking about the science. I like figuring out strategies for experiments. I like figuring out problems, reading the literature, writing scientific ideas. But the actual, being at the thrust of doing the experiment and constantly bumping up against frustration and failure, I actually realized at its core, I just don't like it. And so it was extremely hard. It was extremely hard to be motivated. Um, Extremely hard to feel like it had any meaning or purpose to it.
Hmm.
And it was also complicated by the fact that in December 2020, well, let me back up a little bit. I think Caltech did a very bad job responding to the pandemic for a lot of reasons. One of the reasons was that they started surveillance testing very late. They waited until Thanksgiving of 2020 to do
Oof.
surveillance testing. Um, and I spent a lot of time in the summer of 2020, kind of going into August and September, working with a couple of my friends on a like 40-page report on all the shortcomings that of Caltech relative to its peer institutions. And that really burnt me out.
Hmm.
Um, I was really, prioritizing that over my lab work, honestly. And it was a lot of bashing our heads against the administrative wall of trying to enact some kind of change. And, you know, there were some things that changed, which I'm proud of, but of course it didn't feel like enough. But by the time that Caltech started surveillance testing, they did it very understaffed. And there was a team of nurses at the Caltech Health Center that were working like 14-hour days with no days off
Wow.
for weeks. And at the time, I was getting allergy shots, which is, trying to retrain my immune system to not be allergic to as much pollen and everything else that I'm allergic to. And in December, my the nurse who was administering it was so overworked that she gave me 10 times the dose of all the
Oh
stuff that I'm
no.
allergic to.
Oh no.
Um, and had a severe allergic reaction, had to go to urgent care. People with COVID were going in and out. I was like in an N95 mask with my face swelling, ears swelling. Um,
Oh no.
and then they treated me with steroids, which was great because it was what I needed, but are also immunosuppressants. And so I recovered from this and then came down with COVID pre-vaccine. And I was already planning leave of absence for my mental health, but after this, I needed it for my physical health too. And so I started 2021 just on a leave of absence, and it was so rough. I mean, just when you go through something like this with your institution that you feel like should care about you, but is messing everything up, and you have a bad experience with medical accident, and there are all these feelings of uncertainty and a kind of institutional betrayal that compound with the uncertainty of research, the uncertainty of the whole world, and it was, I mean, excruciatingly painful to just
Wow.
kind of try to cope with it all. And I wasn't sure that I would come back to my PhD. Um, it was, yeah, on the one hand, I kind of felt grateful because, you know, as a PhD student, you have a kind of stability, like you're not paid nearly enough, but that if things are working kind of as they should, you have, 5, 6, 7 years to just be in one place. And that's quite unusual. But yeah, I think I had this kind of spiritual breakdown of all the uncertainty that I was trying to manage. Um,
Wow.
Yeah, and then going back to my immigrant story, I had a conversation with my mom, which was very classic, which was like, "Oh, well, we came to this country so that you could have the highest possible education, so please, for me,
Oh.
finish your PhD."
Oh, that's
Which
so heavy. That's so heavy.
yeah, I still honestly resent. But um, in the end, I decided to come back, and in part this was thanks to my undergrad advisor again, because often when you take a medical leave of absence in the US system, they stop paying you, and you still have rent and
Right.
food and whatever else. And I reached out to Aaron and asked him if I could work part time on the remaining threads from my undergrad work, basically. And he very graciously said yes. And so during this leave of absence, I was doing a lot of bioinformatics. And I remember this, this has happened to me lots of times, and probably other people who deal with depression have experienced this too. But this kind of almost modular regaining of my personality, where I remember a day where I felt a spark of curiosity again for the
Hmm.
first time. And
Wow.
then a day when I felt like, oh, I can focus on something for more than 20 minutes. And then, kind of like a steady rebuilding of myself. And by the time I was done with my leave of absence, I felt like, yeah, I can try again, and if it doesn't work out, I'll leave the PhD program, but at least I'll give it a shot. And that's where I really also appreciate my PhD advisor, because she had enough trust and patience in me to just be like, yeah, explore these ideas, try them out will like find a way for you to be able to finish. And that degree of trust, and confidence was very relieving because I didn't feel pressure from her to accomplish anything specific, but rather just try to find my path back. And yeah, and so like things really started coming together again in like 2022. Um, got the genetics working, and yeah, ended up finishing. But it definitely didn't feel like a done deal for a lot of it.
Wow. Yeah. I'm assuming the sense of closure was far out of reach in that sense, right? Yeah.
Yeah.
Wow. Now, while the ink was drying on your thesis, were there any moments that you were really looking back on and in shock or surprise at how far you've gotten? Like, was there a moment of
Yeah.
realization of just like, wow, I'm an expert at this, in spite and despite of all of that?
Yeah. There are in a couple different ways. It does, it does feel really good when you finally ironed out your protocols and the experimental framework and the data analysis and how to interpret your results. And then you can just kind of relax and do the work.
Mhm.
And a lot of people have said this to me, and I, more or less agree. It's kind of funny because by the time that I was done by my PhD, I felt that if I knew what I know now, I could probably do the whole thing in like 6 months, 9 months.
Oh,
It's kind of like once everything's established and it works,
yeah.
in a sense, it's not that hard. The hard part is figuring it all out. And so I did feel a kind of expertise. One funny moment was I came back after my leave of absence and I had a bunch of media bottles. And I looked at them and they weren't contaminated. I was like, oh, I can keep things sterile.
Yeah, just ultimate
I
validation. Yeah.
Yeah, I can be away for 5 months and it's still fine.
Oh, nice, nice. No,
um,
that's important. That's important.
yeah. Um, so there were some little things like that. Uh, and then about the thesis itself, I I had this consternation about my acknowledgement section
Hmm.
because I found myself, trying to express all the injustices that I felt I witnessed during my PhD. And so I ended up writing a separate gratitude section and a separate acknowledgement section where in the acknowledgements one, I was basically writing about everything that I felt was wrong. And in the gratitudes one, I was actually thanking people. Um, and that gave me a sense of closure. I think just like exerting the agency that I had over my thesis, where nobody can tell me, what I
Hmm.
can or can't write,
Yeah.
and finding a way of expressing myself felt really good. Um,
Well,
yeah,
it's like a protest by publication. That's
yeah,
great.
exactly.
Very good.
Um, yeah, I also, I mean, the past however many years have been wild. I ended up actually writing my thesis, and it was possible to do this because I had incremental publications during my PhD rather than waiting for one big one. Um, I ended up actually writing the whole thing in like 3 weeks because uh,
Wow.
we had a dear friend of ours who was escaping like an abusive situation and facing homelessness who was living with us, and like he had a cat, we had cats that weren't getting along really, and then got COVID again, and like all these things that like in the buildup of when I should have really been wrapping up and graduate working on my PhD and thesis, it just was so much life stuff was happening that I ended up having to write the thesis as kind of stream of consciousness, just like start to finish, which I don't recommend. Um, but if you're in that position, it's possible.
That's advice through suffering. Yeah,
Yeah.
that's real. Um, if you could turn back the hands of time and give your prior self a little bit of advice, what do you think it would have been, and like at what stage do you think it would have been best?
That's a very good question. Um, I'm trying to think of the advice that I give other people because I feel like it's easier to give to listen. It's easier to give other people your advice than to listen to it yourself.
100%.
I think, well, I guess if I knew that the pandemic was going to happen, I think that I would have given myself a lot more advice about pacing and just trying to not push myself as hard. I so what one thing that I try to embody myself, and I've been talking to folks about, is rather than thinking about productive things, to think about constructive things, where sometimes the most constructive thing you can do is take a nap or like hang out with friends and recharge a bit, which isn't going to become, a publication or whatever, but is really going to help you. Um, I think overall, what I'm trying to do now for myself is to figure out how to enjoy the process of things rather than the outcomes. I think that certainly through my PhD and now, so much of my focus has been on trying to get to a specific outcome. And that leads to a lot of goalpost shifting, like this, even this sense of like, well, everything I know how to do is trivial, everything I don't know how to do is impossible. But then when you learn the thing, that becomes trivial and you've moved nowhere. It's like being on a treadmill of like imposter syndrome. But I think that if I started earlier trying to learn how to enjoy the process rather than think about outcomes, I think I would have had a healthier, or if not healthier, just a more sustainable kind of way of moving forward.
Yeah, pace. That's something I need to learn as well. Yeah,
Yeah.
yeah. Um, so, can you tell us a little bit about where you are now and how what particular parts of your training, be it directly or indirectly, or is helping you?
Yeah. So now I'm a postdoc. I'm in Ellen Ye's lab at Stanford, and we're squarely in the non-model protist world, doing a lot of cool things with diatoms, actually, mostly. And I'm nearly 2 years through, and I'm actually completely changing my project right now, like basically starting from scratch, because I've been trying to develop a new model system with this alga, and it just wasn't having it. Um, and I, it's a kind of it's a familiar feeling in terms of the frustration and uncertainty of how research might go. Um, but it's also something that I'm deeply appreciative of Ellen for in her management style is she told me relatively recently that she believes in quitting things that aren't working. And that was like the perfect thing that she could have said to me because at the same time it communicates that she doesn't want to kick me out of the lab because my project isn't working, but rather wants me to find something that works for me in the lab. but also that, yeah, sometimes things don't work and that's okay. And that's kind of in the nature of things. And I think it was, this kind of sequence of, I guess, mentorship that I've had of learning a kind of intellectual honesty and curiosity from my undergrad and a kind of patience and trust and confidence from my PhD and postdoc labs that have come together in a very nice way. Uh, so I'm going to try to see if I can eke out a tiny little like resource announcement paper or something from the past 2 years so that there's some record of it. But pretty much starting from scratch. And my intention is to work at a primarily undergrad university, um, in part because I don't really want to like recruit people into an academic system that I have a lot of problems with, but empowering undergrads to do what they want and what they think is right feels much better for me. But the point is that what I think I need for that is to just kind of demonstrate that I have a research program and a research idea that's appropriate for undergrads.
Mhm.
And that's a totally different goal than if I wanted to go to like a elite R1, glamorous institution. And that creates, I think, this possibility for me to practice what I was just telling you of like pacing and focusing on the process, and maintaining that this is my life and I shouldn't be stalling or like delaying whatever else I want from it. So I feel very lucky in that I found a place where that's possible.
Wow, that's huge. The vision you have of working at a PUI. Can you tell us a little bit more about, in your ideal world, like what would that program look like that you'd want to run?
Yeah. So I've been thinking about this a lot actually because I'm working on a fellowship application right now where I'm proposing some stuff. So I want to do research. I want to teach, and I want those things to be in a kind of concert with each other. Uh, but I also am becoming more and more interested in science education research,
Hmm.
and just the idea of how how does someone's identity as a scientist or as a discoverer develop? And I'm working with someone at San Francisco State University right now, Professor Kimberly Tanner, whose whole career is about this. And in the ideal world, I want to develop an educational program that's modeled after two that already exist. One's called C-phages, which is about finding, phage from the bacteriophages from the soil environment, and one called Tiny Earth, which is about discovering new antibiotics from soil environment. So the first one, they're both based out of universities. They both have international reach, and it's all about working with high school students, undergrads, to get them out there and do actual research in a systematized way. And I want to do something very similar with eukaryotes. With this, so you've probably seen this in the popular science news, but people are talking about more and more of these nitrogen-fixing eukaryotes that have endosymbionts that are akin to mitochondria and chloroplasts, and that's actually what Ellen's lab studies in diatoms. Um, and getting those out of the environment is relatively straightforward. You take samples of sediments from, aquatic systems, don't give them any nitrogen source, and see what grows up. And so
Hmm.
I would love to have this educational program where we do this, modeled after C-phage's Tiny Earth, but also actually use it to research how students develop an identity uh, of being a scientist. Um, and I think there's a lot to learn there, and a lot of ways to improve science education. Yeah, one of the things that this, I guess, is motivated by is there's a fair amount of research that people develop their identity as readers around the fourth grade, and that's kind
Hmm.
of where they decide, am I, I'm a reader, I'm not a reader. And there's research about how you can actually shape that and catch kids before they develop that identity and support it. And I think that we could find something very similar with science. It'll be very interesting to look into.
Oh, that's awesome. That's such a cool initiative. Do you think that this could be possible as like an introduction at the high school level?
I think so. I mean, it really. So what I want to avoid is being one of the well-meaning people who comes to schools and be like, I think you should do this, when the educators have had decades of experience and know like what works and what doesn't. And so I want to be very careful about the claims that I'm making. But the way that I'm envisioning it right now is, do a little pilot study with undergrads, develop the protocols, do, a very careful, rigorous analysis of their sentiment and kind of identity constructs. If it seems promising, steadily develop it until it's systematized and reproducible, and after that, uh, expand to working with high school students.
So cool.
But yeah.
Wow. Um, so with the last few minutes remaining, is there anything you'd like to
Yeah.
plug or pitch, or like the floor is yours? You could talk about whatever you'd like. I'm curious.
Oh wow. Well, I don't have anything to plug or pitch in that I'm not a creator influencer of any particular
It could be a
type.
concept, an idea, something you want to
Yeah,
champion. Yeah.
yeah. You know, all I'll say is I hope that anyone listening to this finds a way to be gentle with themselves and is able to find what gives them a sense of meaning and fulfillment, uh, and the opportunity to pursue that. So I'll express a wish.
That's
Yeah,
wonderful. Thank you so much, Lev. This was really, really cool. I hope you listening at home are enjoying this as much as I have. All right.
yeah.
Thank you
Thank,
very
thank you,
much.
Sebastian. It was wonderful to talk with you.
No problem.